Chimeric antigen receptor T cells. How chimeric antigen receptors help T cells recognize and kill CD19 positive cancer cells, by Rachel Mund and Annie Hutchinson.
Cancer affects almost 40% of our population. For our presentation, we are looking at the experience of a fictional patient, John Doe, who is suffering from non-Hodgkin's lymphoma. We will be discussing how we can treat his cancer using CAR T cells and the amazing way that CAR T cells go about fighting cancer.
Previously, available treatments before the development of CAR T cells had severe side effects and lower efficacy. These included chemotherapy, radiation therapy, and surgery. Lucky for John, he is able to receive CAR T immunotherapy.
To begin, John will get blood drawn and his T cells, CD4 or CD8, will be isolated and grown in a lab. Using these T cells, CAR T cell therapy will be employed to help treat John.
Chimeric antigen receptors, or CARs, are synthesized structures made to target antigens in the body that our T cells otherwise would not be able to recognize. CARs have three main domains: the endodomain, the transmembrane domain, and the ectodomain.
The ectodomain is comprised of a single-chain variable fragment, scFv, and a spacer, also referred to as a hinge. Although there are many different scFvs, we are focusing on anti-CD19. This scFv is able to recognize malignant B cells, which present CD19 proteins on their MHCs. CD19 positive cells in the human body are almost exclusively cancer cells, so being able to target them is a key aspect of the CAR. The transmembrane domain of CARs contains a hydrophobic alpha helix and spans the entire region of the cell membrane. It therefore allows for signaling between the extracellular domain and the intracellular domain. Although initially the transmembrane domain was largely ignored, it is now understood to be an important component of the CAR, since it allows the CAR to bind cell surfaces and therefore helps to control how much signal is present in the cell.
The endodomain consists of two parts, the costimulatory domain and the CD3 zeta. The costim domain can be made from CD4, CD8, or CD28 proteins. The CD3 zeta is responsible for the proliferation of the signal and sending it down the signal cascade. It will eventually cause cytokines to be released.
There are three generations of CAR T cells. The first lacked any costimulatory domains, and in the following generations, they were introduced to enhance proliferation and to direct the expansion of T cells on repeated exposure to the antigen.
There are a few ways to transfect T cells with CARs. There are mechanical and chemical methods, but the most commonly used one is using a lentivirus. The lentiviruses are made by putting viral components, such as the viral envelope, genetic material, and cytoskeleton components into human embryonic kidney cells and grown. Once mature, the virus is titered, and the T cells are transfected with the lentivirus.
After this is done, John's T cells will be activated and cultured. He will receive chemo to wipe out his immune system, and then will receive an infusion of his modified CAR T cells. Antigens present on the MHC complexes of malignant T cells and the CARs bind to those antigens. This triggers the tyrosines attached to the endodomain to be phosphorylated. Then there is a cascade of phosphorylation. This leads to a release of calcium from the endoplasmic reticulum. The NFAT gene is transcribed in reaction to this signal. The resulting mRNA is translated into proteins in the ribosome, where it also binds to transcription factors. The final proteins are expressed and are able to cause the release of cytokines, which will attack the malignant B cells.
CAR T cells are able to specifically target cancer cells through their specific antigens and are able to minimize side effects presented by other cancer treatments. It serves as a novel treatment and a cure for blood cancers with efficacy that hasn't been seen with any other treatments. There is the possibility of CAR T cells eventually being used to attack solid tumors in addition to being able to destroy cancers in the blood. Further research is looking at combining CAR T therapy with other cancer treatments in order to create more long-lasting effects. A study done by Koch and Durfer treated 15 patients with B-cell malignancies with CAR T therapy and found that 8 achieved full remission, 4 partial remission, 1 saw no positive benefits, and 2 patients were unable to be evaluated. There are countless more studies which have been done displaying similar great results. This is a way to fight blood cancers and a novel option to specifically attack tumor cells without affecting healthy cells. Possible side effects include cytokine release syndrome, basal signaling leading to overactivation, and reduced immune system. Future research will lead to trying to find ways to use CARs to fight solid tumors and other types of pathogenic diseases. Many pharmaceutical companies shown are actively researching ways to further this treatment.